This lecture is to give you a brief overview of some of our first year lecture content. And specifically, I'm going to focus on a few slides looking at microorganisms. Here you can see a prokaryotic cell. And in general, a prokaryotic cell would be something that is typically unicellular and can only be seen at a microscopic level. Now, there are two main types. There is the archaeobacteria, and these are primitive types of prokaryotes. So they live in volcanoes and geysers and salt lakes, so very hostile, extreme environments. But there are also bacteria, which is something we're all familiar with. Here you can see the prokaryotic structure, and some of the main differences you see between prokaryote structure and eukaryote is the lack of membrane-bound organelles. So in our human cells, we would have organelles such as the endoplasmic reticulum, the Golgi apparatus, etc. And they had these phospholipid membranes. But you don't see these in the prokaryotic cells. So for instance, they don't have a true nucleus, which is bound by a nuclear envelope. They have their genetic material diffusely spread within the cytoplasm in an area called the nucleoid. They do have their own ribosomes, which you can see with here with the little red dots, and that enables them to make their own proteins. But they also have some distinct structures. So we can see these fimbriae, hair-like projections on the outside, and this helps them to stick to surfaces, which is very important if they want to invade a host, for instance. And also, we've got this larger hair-like structure known as the pilus. And this is a structure that allows transfer of DNA from one bacterium to another by cell-to-cell contact. And lastly, this large flagellum, and that's used to be able to move that bacterium towards or away from something. So for instance, it has a chemotaxis response, and that might be a positive chemotaxis response towards nutrients and oxygens and things that it needs, or it might be a negative one so that it can actually evade toxic substances. What I also want to draw your attention to on the outside is that we have the plasma membrane, cell wall, and the capsule. And we'll talk about the cell wall at another time, but it's really important because the structure of that cell wall can make a difference when it comes to treating bacteria that are harmful. As we know, bacteria are something that can be very good. They can help decompose dead matter. They can also provide vitamins within our gastrointestinal tract and prevent pathogen colonies forming there. However, we also know that they can be our foe. They can lead to things such as food poisoning, septicemia, and opportunistic infections. One way of being able to classify bacteria is according to their cell wall. So we have these gram-positive purple stained bacteria, and they have this thick peptidoglycan cell wall, and we have these gram-negatively stained bacteria which have this pink colour, and they have only a very thin layer of this peptidoglycan. It's important to be able to differentiate between the two types of bacterium because they'll be responsive to different types of antibiotics, and you need to ensure that any patient would have effective antibiotic treatment. Anyway, this stain works as follows. So we apply on the left in point 1 a dye known as crystal violet, and it has this purple colour. We next apply iodine, and iodine acts as a mordant. So in this case, it binds to the crystal violet and forms a bond between it. And it creates this very large crystal violet iodine complex. And you can see there that all of those bacterium are stained purple. We then rinse the sample as seen in point 3 and we get some decolorization. So the bacterium with the thick peptidoglycan walls will trap in that violet dye, and you can see here that we have these round cocci that are stained purple. We then stain with a red dye known as safranin, and this can enter the cell and bind to DNA. And this leads to a pink coloration. So here you can see two different types and populations of bacterium according to their cell wall. As well as being able to identify bacteria according to their cell wall, we can also define them via their shape and their formations. So for instance, in the purple, we can see here on the left a coccus that has this round spherical shape. And as we move across to the right, you can see we have two, so that's a diplococcus. Sometimes it's also known as a diplococci. So cocci is where there is more than one of those spheres. We have the 4 with the tetracoccus, this grape-like formation with the staphylococcus, and this filament-like formation with the streptococcus. If we now look in the center in blue, on the left we have this rod-shaped bacillus. Again, that's the singular term. It can be known as bacilli when there is more than one. We have these diplobacillus where we have 2. We can also have palisades where you have this stacking formation of the bacillus. And again, this filamentous type and form, which is the streptobacillus. And then there are also some other types of prokaryotes which have quite different structures. So we have these vibrios, which have this comma shape, and we have these spirochetes, which have this sort of helical shape. So we can identify the different types of bacteria according to this shape. And this then gives us an idea of what that bacteria is. So I've included some examples here. You can see for the bacillus, a common example that we'll all be aware of is E. coli. And we'll have some harmful E. coli that we're aware of. There are other things again, such as the Staphylococcus aureus, the Streptococcus pneumoniae, and you can see down on the very bottom left, the Vibrio cholerae. Now that we've looked at the gram staining and we've looked at some different shapes, you should now be able to define the different types of bacteria that's in these panels. So have a look and see if you can identify them according to their gram staining and their shape. Okay, so now let's start off by looking at the gram staining. So this top left image, do you think that this is gram positive or gram negative? I'm going to give you 10 seconds to just pause this recording and then to come up with an answer. It is gram positive. So you can see here it has this purple staining where the crystal violet dye has been trapped within that bacteria. Now if we look at the top right image, is that gram negative or gram positive? And again, I'll pause for 10 seconds for you to be able to answer this. This one is gram negative. So you can see here this has this pink colour and the crystal violet has been lost because it has this thinner peptidoglycan wall. How about now the bottom left image? This one is also gram positive. And again, you can see that pink coloration. And then finally, the bottom right image, is that gram-positive or negative? That's gram-positive because you can see this purple staining. Now we're going to look at the shapes of these particular cultures. So again, if we look at the top left image, what shape is that? Again, I'll give you 10 seconds to have a think about it. That is bacilli. So as you can see here, we've got these rod-shaped bacterium, and because we've got more than one, the plural is bacilli. How about the top right image? And again, I'll give you 10 seconds to think about this. This is also bacilli. So you can see it looks a little bit smaller, but again, you can see this characteristic rod shape present there. How about the bottom left image? Well, in this case, we have cocci. So you can see in this one that we have these spherical or round-shaped bacterium there. And then finally, on the bottom right, what shape are these? These are also cocci, and you can see that these have this lovely defined round shape there. So lastly, I would like to say thank you very much for listening, and I hope you've enjoyed this short lecture and learned a little bit more about prokaryotes. Thank you. Bye-bye.